चलो गाइस डीएनए फिंगरप्रिंटिंग का एनसीईआरटी रीडिंग करते हैं।
5.10 DNA fingerprinting. As stated in the preceding section, 99.9% of base sequence among humans is the same. Assuming human genome as 3 times 109 bp, in how many base sequences would there be differences? It is these differences in sequence of DNA which make every individual unique in their phenotypic appearance. If one aims to find out genetic differences between two individuals or among individuals of a population, sequencing the DNA every time would be a daunting and expensive task. Imagine trying to compare two sets of 3 times 106 base pairs. DNA fingerprinting is a very quick way to compare the DNA sequences of any two individuals. DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called as repetitive DNA, because in these sequences, a small stretch of DNA is repeated many times. These repetitive DNA are separated from bulk genomic DNA as different peaks during density gradient centrifugation. The bulk DNA forms a major peak and the other small peaks are referred to as satellite DNA. Depending on base composition, A, T-rich or GC-rich, length of segment, and number of repetitive units, the satellite DNA is classified into many categories, such as microsatellites, mini-satellites etc. These sequences normally do not code for any proteins, but they form a large portion of human genome. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA from every tissue, such as blood, hair follicle, skin, bone, saliva, sperm etc., from an individual show the same degree of polymorphism, they become very useful identification tool in forensic applications. Further, as the polymorphisms are inheritable from parents to children, DNA fingerprinting is the basis of paternity testing, in case of disputes.
ऐसे ही वीडियो और शॉर्ट्स देखने के लिए सब्सक्राइब करो बायोटिप्स नीट को।